Hello, this is talk number 28 in a series of Principles of Biochemistry by Albert Lehninger, 7th edition. Today we will discuss the biosynthesis of starch, sucrose, and cellulose, and then continue with a discussion of lipid synthesis. The biosynthesis of starch, sucrose, and cellulose is similar to that of glycogen synthesis. ADP glucose precursors are transferred to starch by starch synthase. Branching enzymes convert the starch to amylopectin. UDP glucose is combined with fructose 6-phosphate to form sucrose 6-phosphate. This is an example of UDP glucose being with fructose 6-phosphate. UDP is right here, and the hydrolysis of this the elimination of UDP and the hydroxyl group causes an ester linkage between the number 1 glucose and number 2 atom of fructose, and this is sucrose 6-phosphate. There's a phosphate, and that is hydrolyzed, and you get sucrose, which is common table sugar. This is a highly regulated process. There is a balance between energy production and storage needs. Enzymes are regulated by dark and light, which respectively stimulate glycolysis and gluconeogenesis. In seed generation, the fatty acids are converted to glucose through glyoxylate cycle before photosynthetic capacity is reached. Succinate is formed from amino acids and enters the citric acid cycle in the mitochondrion. Cellulose synthesis, this is a constituent of plant cell walls. This is a linear polymer of beta-1,4 linked D-glucose units. So sucrose plus UDP gives UDP glucose plus fructose. UDP glucose is added to cellulose, and the plants possess their own metabolic pathways and those of animals. In lipid biosynthesis, we have biosynthesis of fatty acids and eicosanoids. The precursor to fatty acid synthesis is a 3-carbon malonyl-CoA formed from acetyl-CoA and bicarbonate. This is a biotin carrier protein. So this is a representation of that structure right here. And looking at this, you have biotin arm is present here with the lysine side chain, the biotin carrier protein. We have bicarbonate with consumption of a molecule of ADP. We get the bicarbonate attached to the biotin moiety here, and the biotin carrier protein is positioned between transcarboxylase and biotin carboxylase in a linear form. This is the carbonyl group right there, and then finally you get a swinging of the chain from biotin carboxylase to transcarboxylase, and acetyl-CoA is brought in, and this forms an adduct with the carbon dioxide, getting malonyl-CoA and liberation of the biotin moiety with the biotin carrier protein here. Fatty acid synthase, there are 2 types in nature, types 1 and 2, and you have the addition of 2 carbon units. The initiation is as follows. So you have the malonyl group right here with elimination of CO2. This is the breakage of this bond right here, and it attaches to a carbonyl moiety in the first acyl group right here. So this is acetyl-CoA, and the fatty acid synthase is right here, and it is connected through a sulfur bond to this carbonyl right here in the methyl group. So you get elimination of carbon dioxide and the liberation of and condensation, and you finally get production of the growing chain right here with 4 amino acids attached to the sulfur group. This material is then reduced, and the product is right here in intermediate form, and this undergoes spontaneous dehydration right there, and you come up with this structure right here, which is alkene linkage. The hot water is removed from these two spots right here, and then you get another reduction process here, and you get saturation of the growing group with the carbonyl still attached to the sulfur. So you lengthen it by 2 carbons, and you can propagate the process through this mechanism up here. It can go, there's a 4-step sequence for each 2 carbons that are added, beginning with, and so this is how carbon dioxide is assimilated into the process. This is a propagation step. Again, you have fatty acid synthase right here. This is the addition of the 2 carbon unit. There's a reduction process with liberation of this carbon dioxide here. And the propagation continues with elimination of carbon dioxide. And it grows by 2 carbon units with the this sulfur anchoring fatty acid synthase right here. And it grows carbon units, and the process requires 4 reducing equivalents in sequence, and you're relying on oxidation at this point and cleavage of the moiety there. The mechanism is present here. There's a butyryl group shown in this representation here, attached by the sulfur. And then malonyl-CoA with dissolution of this bond right here. There is a reaction that occurs where the carbon dioxide from the malonyl-CoA is liberated. This bond then kicks in and reclaims the sulfhydryl group, and you get condensation. And this then there's a redox reaction. At this point, elimination of water, you get reduction of this carbon right here to a methylene group. The acyl carrier protein holds the fatty acyl chain, the fatty acid synthase. For palmitate, which is 16 carbon fatty acid, you require 7 ATPs, 14 NADHs. The synthesis occurs in animal cytoplasm or in plant chloroplast. In animals, acetyl-CoA has a transport mechanism from mitochondria to cytosol. The acetyl-CoA is transported, combined with oxaloacetate to form citrate. Acetyl-CoA is reformed after the citrate crosses the membrane. Oxaloacetate product is reduced to malate, and then it re-enters the mitochondria, as shown in the previous slide. The regulation of acetyl-CoA carboxylase is a rate-limiting step. Palmitoyl-CoA is a feedback inhibitor, and the enzyme is inactivated by phosphorylation. The acetyl-CoA carboxylase of plants and bacteria is activated by increased pH and magnesium, which require light. Malonyl-CoA blocks beta oxidation and allows fatty acid synthesis. Palmitate, the 16 carbon, is a precursor for longer chain fatty acids in the omega-3 and omega-6 pathways in non-mammals. Recall that this creates double bonds and or a process of removing carbons from the opposite end of the molecule, the non-carboxyl end. Alpha-linolenate, an omega-3 fatty acid, and linoleate, omega-6 fatty acid, are essential fatty acids, and they are obtained in the diet. Eicosanoids are short-range messenger functions. Arachidonic acid is a precursor for prostaglandins, which mediate gastric mucin production or mediation of inflammation, pain, and fever. Aspirin, ibuprofen, and naproxen all are inhibitors of prostaglandin formation. So here's an example of COX, which is a prostaglandin H2 synthase, serine, which has the hydrogen hydroxyl moiety as part of the structure, and you get acetylated inactivated prostaglandin H. This is aspirin right here, and salicylate, ibuprofen, and naproxen. Prostaglandins are converted to thromboxane, which causes platelet clotting. Arachidonic acids can be converted to leukotrienes, which are involved in early steps of the inflammatory response. Triacylglycerols are the storage energy form. Glycogen supplies the energy needs for 12 hours. Stored fat can supply energy needs for 12 weeks. And the same precursors can form glycerol from fat stores or phospholipids from membrane synthesis. Glycerol biosynthesis is stimulated by dietary carbohydrates and fats by insulin, and in diabetes, carbohydrates and proteins are catabolized or broken down to acetyl-CoA and then converted to ketone bodies. About 75% of lipolysis products are reesterified to triacylglycerol, reclaimed and recycled even in starvation states. And the energy produced from glycerol neogenesis is an abbreviated form of gluconeogenesis. So pyruvate is converted to oxaloacetate with pyruvate carboxylase, oxaloacylate to phosphoenolpyruvate by PEP carboxykinase. There's a multi-step process to obtain dihydroxyacetone phosphate. Another enzyme, glycerol 3-phosphate dehydrogenase, then produces glycerol 3-phosphate, and using the esterification of these moieties, hydroxyl moieties, and the phosphate, here you can get triacylglycerol synthesis, where this is the backbone structure and the fatty chains are attached to these 3 carbon moieties. Phosphoenolpyruvate kinase is the rate-limiting step. It is inhibited by glucocorticoids. Membrane phospholipid biosynthesis occurs in the smooth endoplasmic reticulum in eukaryotes and mitochondria. There are 4 steps: the backbone molecule, glycerol or sphingosine, fatty acid attachment, a hydrophilic head group added, that would be at the end, and then the head group is modified. The head modification of the head group occurs for more specific forms of phospholipids. So you get phosphatidylglycerol, cardiolipin, phosphatidylinositol are all derived from CDP diacylglycerol. So here's CDP diacylglycerol, cytosine, ribose. Here's the 2 phosphates. This is a glycerol background with the various R constituent groups of fatty acids are attached here. In eukaryotes, you have cardiolipin synthase. Phosphatidylglycerol can replace the phosphate with liberation of CMP, and alternatively, you can have a phosphorus synthase with inositol replacing, so you can get phosphatidylinositol here with possible phosphorylation. Phosphatidylcholine is lecithin, and this is formed from phosphatidylethynolamine from the methylation from S-adenosyl methionine. Sphingolipid synthesis is in 4 steps. There's an 18-carbon sphingonine from palmitoyl-CoA and serine. A fatty acid transfer to form N-acetylsphingonine Desaturation of sphingosine to form N-acylsphingosine, and phosphatidylcholine is attached. The sphingosine and head group sugar are attached to form glycolipids, cerebrosides, and gangliosides, and polar lipids are transported to the membrane from the Golgi complex, and this originates in the endoplasmic reticulum. And that'll be it for our talk. Thank you.